Good morning, my dear students. Today I am going to teach you about the monohybrid cross, rest of the concept, and test cross. So, Punnett Square. Punnett Square was developed by British genetist Reginald C. Punnett. The production of gametes by the parents, the formation of the zygotes, the F1 and F2 plants can be understood from a diagram called Punnett Square. So this is a Punnett Square, the diagrammatic representation, the gametes, the parental plants. In monohybrid cross, inheritance of one gene, the plants, pea plants, crossed based on one contrasting traits, that is regarding height of the stem. One of the pea plant is having tall stem and another pea plant is having dwarf stem. If these plants are crossed, the one of the tall parental plant is produces the gametes, the capital T gametes, and another dwarf pea plant is produces the gametes containing small t. So capital T, small t, union of the gametes. Here the progeny consists of capital T, small t, genotype. Another progeny is consist of the genotype capital T, small t. Rest of the one also capital T, small t, capital T, small t. So all the progeny plants are consist of genotype capital T, small t in F1 generation. Whereas in F2 generation, here the plants are produces the gametes, capital T, small t gametes, this parental plant. That another parental plant is produces gametes, capital T, small t. So union of the gametes, zygote formation, and the genotype of parental plants. So this Punnett square gives information about the formation of the gametes by the parents, union of gametes, and genotype of progeny. That's maybe in F1 or F2. That's it must be remembered, important for objective point of view. Who proposed the Punnett square? Reginald C. Punnett. Its Punnett square is also known as checkerboard. Okay. So Punnett square or checkerboard gives the information about the production of gametes by the parents, the formation of the zygotes, a union of the gametes, and also the genotype of progenies, or F1, F2, F3 or F4. The Punnett square shows the parental tall capital T male and dwarf female plants. The gametes produced by them and the F1 capital T small t progeny. Okay, so F1 progeny, if the tall parental plant is crossed with a dwarf parental pea plant, the F1 progeny form. The F1 progeny is consist of genotype capital T small t. The F1 plants of genotype capital T small t, when self-pollinated, produces gametes of the genotype capital T and small t in equal proportion. When fertilization takes place, the pollen grains of the genotype capital T as well as the of genotype small t. Also, pollen grains of genotype small t have a 50% chance of pollinating eggs of genotype capital T as well as of genotype small t. As a result of random fertilization, the resultant zygotes can be of the genotypes capital T, capital T, small t, or small t. From the Punnett square, it is easily seen that 1/4 of the random fertilization leads to capital T, 1/2 leads to capital T, small t, and 1/4 to small t. Though the F1 have a genotype of capital T small t, but the phenotypic character seen is tall. At the F2, three-fourths of the plants are tall, where some of them are capital T, while others are capital T small t. Externally, it is not possible to distinguish between the plants with the genotypes capital T and is capital T small t. Hence, within the genotypic pair, capital T small t only one character, the T capital T tall is expressed. Hence, the character tall is said to dominate over the other allele, this small t or dwarf character. It is thus due to this dominance of character over the other that all the F1 are tall, though the genotype is capital T small t. And in the F2, three-fourths of the plants are tall, though the genotypically 1 by 2 are capital T small t and only 1/4 are capital T capital T. This leads to a phenotypic ratio of three-fourths tall 1 by 4th capital T and of 1 by 2 capital T small t and 1/4 small t. That's 3 is to 1 ratio, phenotypic ratio, but genotypic ratio of 1 is to 2 is to 1. So the 1/4 is to 1 by 2 is to 1/4 ratio of capital T 1/4 capital T small t of 1 by 2 and 1/4 small t is mathematically condensed to the form of the binomial expression, that's ax plus by whole to the power of square, that has the gametes pairing genes capital T or small t in equal frequency of 1 by 2. The expression is expanded as given below, that's 1 by 2 capital T plus 1 by 2 small t whole to the power of square is equal to 1 by 2 capital T plus 1 by 2 small t into 1 by 2 capital T plus 1 by 2 small t is equal to 1 by 4 capital T 1 by 2 capital T small t plus 1 by 4 capital small t. Now we have to know about the test cross. Mendel crossed the tall plant from F2 with a dwarf plant. It's known as test cross. So the F2 generation plants are crossed with dwarf plant or recessive parental plant is known as test cross. It is used to determine the genotype of a tall plant at F2. Here in monohybrid cross, tall pea plant crossed with a dwarf pea plant, F1 progeny are formed. There the F1 progeny or consist genotype capital T small t. So genotype of F1 progeny capital T small t. Phenotype is tall. That plant progeny plants are produces tall stem. So here the F1 progeny plant F1 generation plants crossed with the recessive parental plant is a test cross. So monohybrid test cross, F1 progeny genotype is capital T small t. The dwarf or recessive parental plant is small t. So here it's the genotype containing capital T small t plant is considered as female, and small t containing plant is considered as male parental plant. So capital T small t containing the female parental plant is produces two types of gametes, that's capital T small t, whereas small t genotype containing parental plant is produces one type of gametes, that's small t. So here the gametic fusion. So capital T small t. Gametes are fused. So progeny here the progeny plants are phenotypically tall and heterozygous tall parental plant, Whereas for another progeny, the small t containing gametes are fused to form progeny. Here the progeny is consists genotype small t. Phenotypic character is dwarf homozygous pea plant. So what is the phenotypic ratio? Phenotypic ratio, the tall heterozygous 50%, dwarf homozygous 50%. So the ratio, phenotypic ratio is 1 is to 1. Whereas genotypic ratio, the containing capital T small t containing 50% to small t containing 50%. So the ratio, genotypic ratio of test cross in monohybrid cross is 1 is to 1. So both phenotypic and genotypic ratios are similar.